Good afternoon, everyone. I'm Dr. Shirin Rana, a postgraduate resident in Department of Pediatrics, Maulana Azad Medical College, New Delhi. In today's presentation, we will be discussing regarding dengue vaccines under the expert guidance of Dr. Anurag Agarwal, Professor, Department of Pediatrics, MAMC, Delhi, reviewed by our expert, Dr. Sunil Kumar Agarwal, Professor, Pediatrics, SCB MCH and SVPP GIP, Cuttack. Talking about the epidemiology, there are hundreds and millions of dengue infections which occur annually, with an estimated 96 million people showing clinical symptoms. Out of these affected people, dengue related deaths range from 5,000 to 40,000 per year, and predominantly these deaths affect children. There has been a trend in fatalities that the deaths have increased from approximately 28,000 reported in 1990 to approximately 36,000 deaths reported in 2019. There has been a shift in the age impact in 2020, as earlier less than 5-year-olds were affected more in dengue related deaths as well as the DALIs. However, now it is seen that aged 15 to 49 years are affected more. The regional impact is such that Southeast Asia and South Asia have borne the highest dengue burden from 1990 to 2020. Talking about the Indian scenario, in 2022 dengue census, it was seen that India reported approximately 1.1 lakh cases from January to October, which is similar to that reported in 2018. The impact of COVID 19 was seen as during the COVID 19 pandemic, the dengue cases dropped by 56 to 60%. The seroprevalence of dengue fever is that in India, approximately 56.9% seroprevalence prevails, which is based on 7 studies. Out of these seroprevalent cases, the symptomatic incidence occurs in 35 to 60 per thousand person years across regions. There are some data gaps such that a systematic review emphasizes the lack of population based data and the need for community studies to estimate dengue burden accurately. There were some challenges experienced during dengue vaccine development. First is that there is existence of 4 types of dengue virus types, that is 1, 2, 3 and 4, and each one of them is capable of causing infection, disease as well as death. There are no validated immune correlate of protection, that is there is no cross protection among these viruses. Animal models do not comprehensively recapitulate the human dengue infection or disease experience. Immunological assays are unable to precisely define the dengue virus type specific, that is homotypic immune responses. And also, there is requirement for very large efficacy trials to demonstrate benefit across diverse populations. Let us talk about the globally available or likely to be available dengue vaccines. First, we'll talk about Dengvaxia. Dengvaxia is a live attenuated tetravalent vaccine. It is a hybrid chimer in which there is backbone of 17D strain of yellow fever virus. Here we remove the pre-membrane protein and the envelope protein genes of yellow fever and replace it with the pre-membrane and envelope genes of dengue virus subtypes 1, 2, 3 and 4. This vaccine does not contain NS protein of dengue virus. It is given as a 3-dose schedule, that is on day 0 at 6 months and the last dose at 12 months, and the vaccine is given via subcutaneous route. The target age group is 9 to 45 years and it depends on the dengue transmission intensity in a given country. Sanofi Pasteur licensed the first dengue vaccine in Mexico in 2015 and more than 20 countries thereafter. It is studied in 26 clinical trials, including more than 41,000 volunteers. CYD 47 was performed in India to assess Dengvaxia's safety and immunogenicity in Indian populations. The WHO says that this vaccine has an efficacy rate of approximately 80% against the disease, hospitalization, and severe disease. Dengvaxia is safe and efficacious in seropositive individuals, but it carries an increased risk of severe dengue in seronegative individuals, that is, those who experience their first natural dengue infection after vaccination. The reason behind this is antibody dependent enhancement. So, if pre vaccination screening is not feasible, vaccination without individual screening could be considered in areas with recent documentation of seroprevalence rates of at least 80% by age 9 years. Screening tests used should be highly specific to avoid vaccinating seronegative individuals and highly sensitive to ensure that all seropositive individuals are vaccinated. In travelers with documented dengue illness in past or are seropositive, vaccination could be considered before travel to high dengue transmission settings. So, to conclude about Dengvaxia, WHO SAGE modified its original endorsement of Dengvaxia, recommending it to be used only in dengue immune individuals. It remains licensed in many countries, including the US. However, Sanofi is going to discontinue Dengvaxia in the third quarter of 2026. Vaccination implementation and uptake has been low, and currently it is not licensed in India. The second vaccine we'll be talking about is TAC-003 Qdenga. It is also a live attenuated, chimeric, tetravalent vaccine with a backbone of wild-type attenuated dengue virus 2, where pre-membrane and envelope genes of dengue virus 1, 3, and 4 are inserted into the backbone. It is given as a 2-dose vaccine on day 0, followed by second dose on 3 months. The age group targeted is 6 to 45 years. However, Qdenga has been studied in age more than 4 years. Regardless of baseline dengue immune status, Qdenga can be administered as there is no fear of antibody-dependent enhancement with it. Qdenga was developed by Takeda. Recently, it has approval from Indonesia, European Commission, and Brazilian regulators for use. Approval was based on safety, immunogenicity, and efficacy data from 19 phase 1, 2, and 3 trials with more than 28,000 participants. Qdenga is authorized in 40 countries, and Indian trials are still going on. The CDC and ACIP recommendations are that Qdenga protected seropositive recipients against all dengue serotypes. Among seronegative recipients, it only protected against dengue virus 1 and 2 infection. However, it did not protect against dengue virus 3. The vaccine's performance against dengue virus 4 infection in seronegative individuals could not be conclusively determined because there was low event numbers, and secondly, there is a lack of a defined immune correlate of protection, made the significance of the immunogenicity data unclear. The third vaccine is Butantan dengue vaccine. The Institute of Butantan, US, NIH, and Merck reported the first results from a phase 3 trial in Brazil with over 16,000 participants and at least 2 years of disease surveillance. It is a single-dose vaccine approved for age 2 to 59 years, and it protects against all the dengue types of any severity. This vaccine is also given regardless of the baseline dengue immune status, and there is no fear of antibody-dependent enhancement. It is a live attenuated vaccine, a tetravalent vaccine, and the backbone is formed by the chimeric dengue virus 2. It is a single dose vaccine, and it was seen that it prevented against dengue virus 1 and dengue virus 2 regardless of dengue serostatus at baseline. There is no data on dengue virus 3 and dengue virus 4. Cases of dengue virus 3 or 4 were not observed during the follow-up period as there was low circulation of these serotypes during trail period. There were no severe cases or cases with clinical warning signs reported. By 21 days after vaccination, only 3, that is less than 0.1%, had serious adverse events related to the immunization. So, summarizing about all the 3 vaccines, all 3 are live attenuated tetravalent vaccines, and they have demonstrated higher efficacy in dengue immune recipients, higher efficacy against more severe clinical phenotypes, variants in dengue virus type specific efficacy, and there is a challenge of capturing data for all desired clinical endpoints, that is any dengue, severe dengue, hospitalized dengue, across all dengue virus types 1 to 4 in both dengue immune and non-immune recipients. This is a table summarizing all the 3 vaccines. The other vaccine candidates are Tdenv-PIV, that is a purified inactivated vaccine. Its phase 1 is completed and phase 2 has begun. Merck's V180, it is a recombinant subunit vaccine, and the phase 1 data is awaited. Then there is Dengue All. Its phase 3 trial was started in August 2024 in India by ICMR and Panacea Biotech. Then there is dengue vaccine from Serum Institute of India, whose phase 1 trial in Australia has been completed, and seroconversion through day 85 ranged from 79.5% for dengue virus 1 to 100% for dengue virus 2. Phase 1 and 2 in India have begun, and it's same NIH vaccine with some variation in formulation. So the perfect dengue vaccine would be one which would protect across all age group. It would prevent infection ideally and disease caused by any dengue virus type, prevent all clinically relevant phenotypes of dengue, not only the severe disease, protect recipients regardless of their immunity status, disrupt transmission of virus between people and mosquitoes, and have durable protection until the recipient is out of the risk window by acquiring a profile of homo and heterotypic immunity, what is observed after 2 natural dengue virus infections. A vaccine which does not increase the recipient's risk of dengue but can reduce the risk of disease or severe disease caused by some of the dengue virus types would still deliver some public health benefit, especially true for dengue virus types 1 and 2. This test and vaccinate strategy, although feasible, could be very difficult to implement across all the dengue endemic areas. So the current status in India is that the Indian trials are going on, and currently no dengue vaccine has been approved for use in India. Thank you.